So the genotype is made up of what we call genes. So genes are the unit of heredity, and it controls the traits that determine the phenotype. So if you have the genes for blue eyes, then your genotype will be blue, or will be the genes that code for blue eyes, and your phenotype will be blue eyes. So seven, locus. Locus is kind of like the location of a particular gene on a chromosome. So, for example, on the Y chromosome, for instance, one of the sex chromosomes, there is a gene called the SRY gene. So the location of the SRY gene on the Y chromosome is that SRY gene's locus. So the next thing that we're talking about is alleles. So alleles are pretty much just versions of a gene, right? So continuing with our eye color example, um, the gene for eye color may be on a certain chromosome, but there's different alleles for that gene. There's an allele for blue eyes, there's an allele for brown eyes, there's an allele for black eyes. So like that, those are different alleles of that eye color gene. So in Mendelian inheritance, which is pretty much what Mendel did, what Mendel saw, what Mendel predicted, there's always two alleles per gene. So for eye color, you might have a brown allele and blue allele. But note, this is not what happens in real life. Often there's multiple genes controlling, a single trait, or there's multiple traits coded by a single gene. So with that in mind, for Mendelian inheritance, always assume that there's two alleles. But in real life, there's usually many more alleles. So next two terms, dominant and recessive. So a dominant allele is an allele that dominates over others. So for example, continuing with our blue eye, brown eye example, let's say the eye color gene has two alleles, blue eyes and brown eyes. If the brown allele is dominant, that means that if you have a single copy of that dominant allele, that dominant brown allele, you will have brown eyes. Even if you have 100 copies of that blue allele, but you just have one copy of the brown eye gene, because that brown eye gene is dominant, that is what will show in the phenotype. The recessive would be the blue allele, and that phenotypic expression is hidden when there is a dominant allele. You don't have to know about hybrids. Okay, so now let's get into Mendel, right? So Mendel, he created three laws, which we will talk about. The first law is the law of segregation. And pretty much the law of segregation states that each parent is going to give you one gene or one allele. And so you will have two alleles. So relating this back to our discussion on the cell cycle with meiosis, you remember meiosis was how we took our cells and we made the germ cells. If you remember, germ cells are haploid, meaning they contain only one piece of DNA, or they contain one set of your chromosomes. So that means when the two germ cells, which is the sperm and the egg, the sperm for males and the egg for females, when they combine to make a zygote, they kind of combine their genes. So that means you get half a set of genes, or one set of genes from your father, one set of genes from the mother, and these will combine to create you, or a diploid organism. So if you remember, all of our somatic cells are diploid, which means we contain two copies of each gene. So any organism, or not any organism, but just us in general, we contain two sets of chromosomes, which means that for each gene, we contain two alleles. So with the law of segregation, your mother gives you one set of chromosomes, and your father gives you one set of chromosomes. So that's the law of segregation. So this is genotype versus phenotype. We already covered that. So, but this is something important about genotypes, how we classify genotypes. They're either homozygous or heterozygous. So homozygous means that those genes with the alleles, or those versions of the alleles. They have the same allele. So going back to our blue eye, brown eye example, if you are homozygous, that means you have two blue allele genes, versions of the gene, or you have two brown alleles. So that is homozygous. You have the same type of allele. Heterozygous means you have different alleles. So with heterozygous, you would contain, or you would have a brown allele and a blue allele for your eye color. So again, going back to our dominant and recessive, we said that brown was dominant. So in heterozygous organisms, the dominant allele will be expressed. That's because, if you have a recessive, it's not going to show over the dominant. So a little bit about notation. When we write, and we're going to do some problems with this, but when we actually write about like alleles and stuff, the dominant alleles are always capitalized and recessive alleles are lowercase. So if it's the eye color gene, that means brown would be capital B, and then blue would be lowercase b. So the rules of probability govern genetic inheritance. You guys don't really need to know about probability because we don't go that deep. But okay, Punnett squares. So Punnett squares are like pretty much the key to Mendelian genetics, and you guys are going to do a lot of problems with Punnett squares. So we're going to go over those. We're going to do a bunch of problems. But some more terminology. A test cross, pretty much when you mate an individual that has a dominant phenotype for a trait with an individual with a recessive phenotype, the genotype of we can figure out the genotype of the dominant parent. This is because, with the phenotype, it's often hard to tell the genotype. And if you think about the reason why for this, well, you can either be homozygous dominant or you can be heterozygous, and you will still express the dominant phenotype. If you're homozygous recessive, which means you have two recessive alleles, you're going to be recessive. If you have two dominant alleles, you're going to have a dominant phenotype. And if you're heterozygous, you're also going to have a dominant phenotype. So if you have two, there's two different genotypes for that dominant phenotype. This means that, we have to determine what that dominant genotype is if somebody has a dominant phenotype. So the way to do that is with a test cross. And we'll get into how those work when we do our questions. And then the other thing is monohybrid cross. So this is just monohybrid, just means that there's only one gene in question. So we're only talking about one gene when we do our Punnett square. So let's look at a Punnett square. Okay, so we're just going to go into questions. So I'll just do this one with you guys because I'll show you how to do it once, and then I guess we'll do it together. So Lisa and Mark are thinking of having a kid, and Lisa is heterozygous for brown eyes, and Mark is homozygous for brown eyes. So what is the probability that their kid has brown eyes? So we want to try and figure out, what the probability of their kid having brown eyes is. So let's think of how we would do this using a Punnett square. So if you draw a Punnett square, it just looks like this, right? It's just a square like that. And the reason we draw it like this is because each parent has two alleles. So let's look at Lisa's alleles. So Lisa's alleles, she's heterozygous, right? So she's going to have one dominant and she's going to have one recessive. Now, Mark is homozygous for brown eyes. Now, because we know that when Lisa, right, Lisa is heterozygous, meaning that she's going to express the dominant phenotype. So that means if Mark is homozygous for brown eyes is the dominant phenotype, because, if Lisa's heterozygous, that means she's going to express the dominant phenotype because she has this capital B here. So that means Mark is going to be capital B, capital B. And he's also going to have the same phenotype as Lisa. So now let's look at how we're going to use the Punnett square. So what you're going to do is you're just going to put Lisa on this side, and you're going to put Mark on this side. So Lisa has a dominant B and a recessive B. Mark has a dominant B and a dominant B. So if you remember from the law of segregation, the Mark is going to pass on one allele, and Lisa is also going to pass on one allele. So that's how this Punnett square is working, right? Like this B allele from Mark is going to move into here, into that square. So is this B allele, right? So those are, that's one different combination. You can also have this B allele move into here, and this B allele move into here. That's another different combination of their kid. Now you can continue with this allele moving here, this allele moving here, and then the final combination is just this allele moving here and this allele moving here. So that's how the Punnett square works. We're just looking at all the different combinations of all the different alleles. So let's fill out the chart now. So here we're going to have capital B, capital B. This is going to be capital B, because remember we're just moving moving them into the box. So this is going to be capital B, lowercase b. This is going to be capital B, lowercase b. So now we have our 4 different results for what our kids could be. These are the 4 different results now. So out of these, how many are going to be brown? So the answer to that is all of them, because all of them have a dominant allele, and we know that brown eyes are the dominant allele. So therefore, all are going to be dominant. So, so if anybody has any questions, then just ask them now, because then we're going to move on to problems, and yeah, you guys are going to try this on your own. Okay, so if there's no questions, then let's try the next one. So Lisa and Mark, again, are thinking of having a kid. Lisa is heterozygous for brown eyes, and Mark is heterozygous for brown eyes. What is the probability that their kid has brown eyes? So I'll give you guys like a few minutes to do this. Yeah. Oh, actually, our meeting time is about to run out. So everybody just log off and then log back on. I'll just quickly end the meeting. Remember to log back on, and then we'll continue with this problem where we left off. So I'll just end the meeting. Just join the meeting again. Okay, so we'll just continue where we left off with this question. That is the question. That. So again, Lisa is heterozygous. Mark is also heterozygous. What is the probability that their kid has brown eyes? I'll give you guys like a minute to do this, and then we will discuss it.
Mommy, next time when you去 Costco， 肯定要买方便面。
哦，方便面不呀？
Okay, so let's quickly go over this. So let's look at Lisa and let's look at Mark and let's look at what their alleles are. So Lisa is heterozygous, so that means she's going to have capital B, lowercase b. Mark is going to be capital B, lowercase b. They're both heterozygous. Now we draw our Punnett square. Fill in the alleles. So Lisa on this side, you can put Mark on this side. Doesn't really matter which side you put them on. You can do either. So Mark is BB, Lisa is BB. So filling in the chart, BB, BB, BB. So now let's look at which of these is going to have brown eyes. So let's think about how we're going to do this, right? So first we need to know which is brown eyes dominant or is it recessive? So brown eyes in this case is going to be dominant. How do we figure that out? Well, I mean, you just look here, right? Because Lisa and Mark both have brown eyes and they have that dominant allele, that means that it's going to be dominant. Brown eyes is going to be dominant. So now, I made a mistake, didn't I? Sorry. This was okay, whatever. So, so we know that brown eyes is going to be whatever is dominant in this case. So B, this is going to be dominant because it has a dominant allele. This is going to be dominant because it has a dominant allele. And this is going to be dominant because it has a dominant allele. So now what is the probability that, you know, it is going to be a dominant allele? Well, it's going to be 3 out of 4 because 3 cases work out of 4. So it's going to be 3 out of 4. So that is how you, again, how you do that. So one more question just to solidify this. So blue eyes is recessive and brown eyes are dominant. John's eyes are blue and Lisa, his wife's eyes, are also blue. What is the probability that their child will have blue eyes? I'll give another minute for this and then we'll go over it. Okay, so again, let's discuss. Just remember, if you're going to attempt these, remember to use the Punnett square. Punnett squares are really, really useful in this. So John's eyes are blue. You know, blue eyes are recessive, so that means he's going to be lowercase b. Lowercase b. Lisa is going to also have blue eyes, so that means her allele is going to be lowercase b, lowercase b. Draw the Punnett square. And just fill it in now, lowercase b, lowercase b. So the combinations are just going to be all of this. So how many of these are recessive? Well, I mean, lowercase b is recessive, so they are all going to be recessive. So that's 4 out of 4 equal to 100%. So again, remember to just look at the phenotypes of parents. Looking at the phenotypes of the parents is really important, and that's going to really help you to get the problem. Just make sure you know which is dominant, which is recessive, and what each parent is, and then it should be pretty easy. Okay, so now let's get into a little bit different type of genetics. So this is not really Mendel's, but you guys still need to know this. So this is X-linked genes. So what this means is if you know about males and females, and you know how they differ, you should know that males will have an X and a Y chromosome, and females have two X chromosomes. And so what this means is that if there is a disorder on the X chromosome, let's just use, well, when we're going to do these problems, what we're going to use to denote, you know, alleles on the X chromosome, we're just going to use XD for recessive, and then XD for dominant. That's how we're going to actually use those. But yeah, so if there's a disorder, that means that males are going to have this chromosome affected, but their Y chromosome is not going to be affected because, note, this is an X-linked. Sorry, that was bad underline. But it's an X-linked disorder. So that means it's only going to appear on the X chromosomes, or it can only be on the X chromosomes. So looking here, this is also an X chromosome. This is also X chromosome. So that means that males, in this case, are what we called hemizygous. And what this means is that they only have one copy of the disease or that chromosome or that allele, because you know they have this Y chromosome, so that's not going to carry any allele from the X chromosome. So that is something, background on how we get these X-linked disorders. So let's go over how we're going to do this right. So Lisa is girl, and then John is going to be boy. So Huntington's disease is an X-linked dominant disease. What this means is that this chromosome, when you have that, or X capital D, is going to contain the disease-causing allele. So if you have a chromosome like this, let's say this is a girl's, genotype. Her phenotype is going to be positive for Huntington's. That was bad. Right? But yeah, so if you have a dominant disease, that means that on the X chromosome, if you have a copy of that allele, you're going to show the disease. So let's look at now the genotypes for each. So Lisa is heterozygous for Huntington. So what does this mean? So let's look at the phenotype. Well, if she's heterozygous, that means she's going to have a dominant and a recessive. She's a girl, which means that, she has two X chromosomes. So she's going to have two X chromosomes. She's heterozygous. She's going to have one dominant and one recessive. So now let's look at John. So John has X, Y, because he's a male. So he's going to only have one chromosome that's going to carry allele. So now let's look at John. John does not have Huntington, which means he does not carry that X-linked dominant allele. So instead, he's going to have a recessive, because if he had the dominant, then he would have Huntington's. Therefore, he must have the recessive. So now, we're just going to use our Punnett square again. So again, we just draw a Punnett square. Now start filling things in. So Lisa is XD and XD. John is XD and Y. So now we just use the same format and we fill in the square. We have XT, XD, XD, Y, XD, Y, and XT, X. So what is the probability that their first child will have Huntington's? So let's look at which of these is going to cause Huntington's. So anything with a dominant allele should cause Huntington's. So this one is going to cause Huntington's because it has a dominant allele. This one is going to cause Huntington's. So that's going to be 2 out of 4, 50 to 50%. That's our answer. We're going to have 50% of our children be dominant. So now I want you guys to try the next one on your own. So hemophilia is an X-linked recessive disorder. Lisa is a carrier and John does not have hemophilia. What is the probability that their son has hemophilia? And for reference, I didn't include this. I forgot to do so. But carrier just means heterozygous. So Lisa is heterozygous. Okay, so let's just go over it now. So this is a little bit of a trick question because there was a little bit more than there seems in this question. Let me just pull my. So let's look at this. It's X-linked recessive. So that means Lisa, I said, was heterozygous or carrier. And so I just want to talk a little bit more about what carrier actually means. So carrier only applies in X-linked recessive disorders. And the reason for this is because Lisa, she's heterozygous, she's going to have XH and then X lowercase h. And what this means is that she's heterozygous and she carries the recessive allele. She doesn't express the recessive phenotype, but she still carries that recessive allele. So we call her a carrier. So now we have Lisa's genotype. So let's look at John's genotype. We know John does not have hemophilia, which means that he must have dominant and Y chromosome because he does not carry hemophilia. Now let's actually draw the Punnett square. So just fill it in. I don't know why. Well, actually, before we fill this all in, I want to just point out something in this question that might have got some of you. I put the word son in there. What is the probability that their son has hemophilia? So what we're actually going to look at is we don't need to look at the daughters. We only need to look at the sons. So how do we determine which are the sons and which are the daughters? Well, the son is going to have a Y chromosome. So we actually don't even need to worry about these boxes because these are not going to have Y chromosomes. So that's a little bit of like a trick that some teachers will pull on you. Make sure you read the question because what is the probability that their son has hemophilia? So they're telling you that they have a son. So now you just have to figure the probability out for the sons. So now we can just fill in the rest of the square, XH and XH. So the chance that their son has hemophilia, we know that it's recessive. That was a horrible circle. We know that it's recessive. So that means that there is a 50% chance, it's 1 out of 2 possible cases, there's a 50% chance that the son has hemophilia. So yeah, if you guys have any questions on Punnett squares, please ask them now. Okay. Let's move on then. So we just talked a lot about Mendelian inheritance. So Mendelian inheritance was that, you have those two genes and all of that, but we didn't actually finish all of the laws, right? We only talked about the law of segregation, which was just that, each parent passes on one allele. So the law of independent assortment. So what is the law of independent assortment? Pretty much the law of independent assortment is that each gene is not what we call linked. So a linked gene is when two genes are on the same chromosome, so they can be inherited together. Now, we're not going to actually talk about gene linkage or anything. You guys don't need to know about that, but just know that with the law of independent assortment, it's that segregation of pairs of alleles are independent of other pairs of alleles. So genes assort independently is pretty much the law. So there's no linkage between any genes. Genes aren't going to be inherited together. Alleles aren't going to be inherited together. That's what Mendelian genetics states. So this is all about genetic linkage. We do not need to cover this. I think, right, this was the sex chromosomes. So sex-linked traits. Okay, they don't actually cover the final law. That's okay. So that was a law. So now let's talk about something. We have to go with the third law. I don't know what I mean. All right, so pull up a graphic here. So the final law, we kind of already covered this, but it's the law of dominance. And this means that some alleles are dominant and they cover up the recessive alleles. Those are the three laws. If you know those, then those can also help you with your Punnett squares and how you're going to make those. So we have to talk about non-Mendelian inheritance. And there's two types that you guys are going to need to know. So one is called incomplete dominance. So this is when the heterozygous genotype creates a completely new phenotype. So look here, we have a dominant or a homozygous dominant genotype. So we get the red flower phenotype. We get the homozygous recessive white phenotype. So we're going to get the white flower. Now, look here. The heterozygous state actually turns into a pink flower. It's actually a blend of the two other homozygous states. And this is what we call incomplete dominance. Incomplete dominance is when you have two alleles when they're in their heterozygous state, they produce an intermediate between the homozygous states. So that's what incomplete dominance is. Now, this is okay. You guys are going to see this a lot when you actually take a bio course or you do any biology exams. The snapdragon plant is the most common example of incomplete dominance. And it has the red flower as one of the homozygous states, the white flower as another one, and the pink flower in between. When you take bio, you will definitely see the snapdragon plant. Now, codominance. Codominance is when the heterozygous state, instead of being blended, you can see both. So like, look, these chickens, I think, they could be roosters, I'm not sure. So the chickens, right, they have a black and white spots on their fur. So this means that they have a white allele and a black allele, and both are being expressed, and they're not being blended. That's the difference between codominance and incomplete dominance. With codominance, there's no blending. It's kind of like you have both are being expressed, but you can clearly see that both are being expressed. There's no actual blending. So that's the difference between codominance and incomplete dominance. So we talked about a common example for incomplete dominance. The most common example for codominance is blood types. So if any of you know about blood types, you know that there's A, B, and the O blood type. But you can have an AB blood type, which means you have an A allele and a B allele. And what you'll actually see on your red blood cells is you will express both the A receptor and the B receptor. That's an example of codominance because you're expressing both, and they're clearly differentiable from each other. Okay, so those are the two types of non-Mendelian inheritance that you guys need to know. So for the last, let's say 15 minutes, we just have a little quiz here. This has some other information that we can go over. So question 1, Gregor Mendel was A, B, or C? I mean, we didn't really cover this. The answer is B, little known Central European monk. Okay, I mean, any of you got that, good job. So which of the following statements is true about Mendel? Again, we didn't cover this, but this is going to teach you an important lesson about all of sciences. If you want to guess, go ahead. If you know the answer, go ahead. So the answer here is actually C, and C is the correct answer. But the thing about, I just want to talk quickly about A. A, his discoveries concerning genetic inheritance were generally accepted by the scientific community when he published them during the mid-19th century. If you ever get a question in any science class saying that somebody's discoveries were accepted as soon as they were published, it's usually false. Almost all scientists face scrutiny after they publish any relatively new or drastic findings. So they're always scrutinized. So if this is an answer choice, it's probably never going to be this. Okay. So now let's get into some actual genetics questions. So Mendel believes that the characteristics of pea plants are determined by the A, B, or C. Okay, so yeah, this is going to be directly from the law of segregation that law of segregation that we talked about. That Mendel's law that each parent is going to donate one allele. So that means that inheritance of units of factors from both parents, so it's definitely going to be A. So an allele is A, B, C, or D. All right, so I'm going kind of quickly, but the answer is D here. It's going to be one of the several possible forms of a gene. Homozygous, remember, that's kind of like a genotype. Heterozygous, it's also, it's specific, like how you inherit those alleles is what homozygous and heterozygous is, and allele is not synonymous for gene. Next question, phenotype refers to the blank of an individual. Okay, again, we're going pretty quickly, but the answer here is B, the actual physical appearance. Remember that phenotype is the physical appearance, and the genotype is the actual genes that you're going to have. Okay, if you have any questions, please just type them in the chat because we can answer them now. So next question, when the genotype consists of a dominant and a recessive allele, the phenotype will be like blank allele. Okay, the answer here is A. Remember that dominant always masks recessive, so the answer is A. Next question, assuming that both parents in the diagram below are homozygous, why would all of the F1 generation have yellow phenotypes? Okay, so this, requires a little bit more of analysis, but let's think about it. So let's look at each of the answer choices and consider if they could be correct. So let's look at A, because the F1 genotypes are homozygous. So if they're homozygous, that means they contain two yellow alleles. But let's actually look at C. Both parents passed on yellow alleles. That's the same thing as A, because remember, if the genotypes are homozygous, they have two yellow alleles. With C, you're going to create homozygous offspring with two yellow alleles, which means that A and C are actually saying the same thing. They can't both be true, so they must both be wrong. So our answer is going to be B. And if you think about B, B is just correct, because if you're crossing a yellow and a green plant and they all produce yellow, then yellow is probably dominant because that is what's showing up in the phenotype. Okay, next question. 8. The idea that different pairs of alleles are passed to offspring independently is Mendel's principle of blank. All right, so the answer here was C, independent assortment. And this is because if you remember back to Mendel's laws, independent assortment means that there's no linkage between the genes, so the alleles are passed on independently. And notice how the question asks different pairs of alleles, which suggests that it's talking about genes, and genes are passed on independently. The law of segregation just means that each parent passes on one allele, and unit inheritance just means, that's not really one of Mendel's laws, so that couldn't be the answer. Okay, in the next question, in the diagram below, what accounts for the green pea seed in the F2 generation? Okay, so the answer here is A. Again, let's just look at the questions. So the yellow allele is dominant over the green one. Yes, that's true, but that doesn't really explain why there's a green seed when we think that there should be yellow. Now, C, the F1 generation parents are homozygous yellow. I don't know why they put parents here. That's a little bit contradictory because they're not really the parent generation. But it should be the F1 generation are just homozygous yellow. So if they're homozygous yellow, that means that they can only pass on homozygous, or they can only pass on yellow alleles, which means that all of the organisms from the F1 generation or all of the organisms in the F2 generation are going to have a yellow allele. That's not the case because we see a green allele, and so the answer is A. Now, this is a correct statement because if you remember back when we did it was the second problem that we did. It was when we had two heterozygous parents. We actually saw that there was a 3/4 chance you're going to get homozygous dominant, which means that complementary probability there's going to be a 1/4 chance that you're going to have it homozygous recessive. So this is the correct answer. The final question, the idea that for any particular trait, the pair of alleles from each parent separate and only one allele from each parent passes to an offspring, is Mendel's principle of okay, the answer here was C. And it seems like this is a difficult concept, just Mendel laws in general for all of you. So let's just go over them once more time before our class ends. Number one, the law of dominance means that some alleles are dominant and they cover up the recessive alleles. Law of segregation, think of like the parents' alleles segregate, they split, they're only going to pass on one. The law of independent assortment means that genes assort independently. Alleles assort independently, which means there's no connection between alleles and genes when they pass on. Okay, so are there any questions of what we covered today? Any questions just in general? Please ask them now. Okay, so if there's no questions, then we'll end the meeting here for today, and I will see you all next week at the same time. Thank you for attending, all of you.